Good morning, viewers. This is Kasturi Day. Welcome to MouShauk. And I'll start with today with seed structure and germination class 9. So I'll be starting this chapter on request of some of my viewers who have requested me to start to discuss some biology chapters of class 9 along with what I am teaching, that is what physics and chemistry, which I am teaching with you all. Okay. So for that reason, I'm starting with the seed structure and germination. This is a concise view of this chapter. As your exams are coming near, so I'll be discussing these chapters in a concise way. But if you want later on details chapter wise, you can request me, or I will discuss you later on when I finish all these chapters, discussing with you all. Then we will discuss the detailed chapters, okay, along with the question answers. In between, if you have any doubt or if you have any questions regarding this chapter, you can comment me in my comment box, or you can also write in the email ID, that is kasturi74@gmail.com. Kasturi kasturi74@gmail.com. You can write there and I will reply you as soon as possible. Okay. So for starting with this chapter, I will request you to please to subscribe my channel if you have not subscribed yet, and ring the notification bell, because as soon as a new video is uploaded, you will get notification. And if you are liking the video, press the like button, share with your friends so that they can also be benefited from these videos. And also do comment and do meditate before you start your day. Okay, so for meditation, let us concentrate on a point of light for just 1 minute. We will meditate and then we will start with the discussion. So first, we'll concentrate on a point of light. Think that point of light is falling on you and your whole body is enlightened. You're getting energy. You're feeling happy. You are tensionless, tension-free, and you are concentrating on your work better. You're happy with your work, you are getting interest in it, and you are doing the job in a very peaceful way. Okay, and at the end of the day, you are seeing that you have become successful, and so you're thanking God for being with you and being at your side. Okay, so we'll end after thanking our God. Okay, so thank you. Thank you for joining, and let us start with the discussion, that is seeds, structure, and germination. So we know that fruit, okay, after the pollination has occurred, the flower gets the pollen grains, that is after pollination, the pollen grains get inside the ovary where there is fusion taking place, and a zygote is formed, which will later form into seed. Okay, and what do you say, that the ovary becomes the fruit, that is the ripened ovary becomes the fruit, and the ovule, which is inside the ovary, that becomes the seed. Okay, the fruit wall, the ovarian wall, that becomes the fruit wall, which encloses the seed. Okay, now what is the function of the fruit? Function of the fruit is it protects the seed and it helps in the dispersal of the seed, so that germination can take place and a new plant can be grown. Okay, for example, mango, pea pod, these all are fruits. Now we come to seed, ripened ovule, the ovule which is inside the fruit, inside the ovary, that becomes the seed. Okay, now the ripened ovule, it contains the embryo. which grows into a new plant. Okay. Now the seed has a seed coat, that is seed wall, you can say, seed cover, which is there, and that has the function to protect the embryo from any mechanical damage. It protects the embryo from any mechanical damage. The seed example is bean seed, peas, etc. Okay. Another thing is there, besides this fruit and the seed, there is another word you will find in this chapter, that is the grain. Now, what is grain? Grain is a fruit. That is also a fruit. But here, the fruit wall and the seed coat, both of them are fused together. In the fruit and the seed, when we talk separately, we will see that the fruit wall and the seed coat, those are separate. Okay, but in this grain, when we are talking about, we will see that the fruit wall and the seed coat, they are fused together, forming a grain. Okay, the function of the grain is forming a protective layer. Okay, for example, maize and wheat. Okay, next. Now, as this chapter is about seeds, so on the basis of cotyledons, the seeds can be classified into two different types, that is monocot and dicot. Monocot means it has one cotyledon and dicot means it has two cotyledons. Okay, the plant, the fruit which has two cotyledons, the seed which has two cotyledons, that is dicot seeds, and the monocot seeds are those which have only one cotyledon in it. And the plant which consists of these monocot seeds, the fruits which are containing these monocot seeds, those are monocot fruits, or the plant which contains this monocot fruits, these are monocotyledon plants. And the plants which contains these dicot seeds, these are dicotyledonous plants. So monocotyledonous plants are coconut, maize, etc. and dicotyledonous plants are pea, beans, etc. Now, another basis of classification of seeds is there. It is on endosperm. How much endosperm is there, or rather what type of endosperm is there, that classifies the seeds into two types, albuminous and exalbuminous. Okay, that is, it contains endosperm. Albuminous contains endosperm, exalbuminous does not contain endosperm. So endospermic and non-endospermic. Okay. Now, albuminous or endospermic, there the endosperm is present. Cotyledon is thin. An example is castor. Okay, and exalbuminous or non-endospermic, that does not contain the endosperm. Cotyledons store food. In albuminous or endospermic, there the cotyledon is thin. But here in exalbuminous or non-endospermic, the cotyledon stores the food. An example is bean. Okay. Now, we know that there is an embryo present in the seed which germinates into a new plant. Now, this embryo has two parts, radicle and the plevule. Radicle, which grows later into root, the root system, and plevule, which later grows into the shoot system. Okay. Now, the radicle, it is the first organ which appears when the seed starts germination. When the seed starts germination, the first organ which appears to us is the radicle. And plumule consists of the short stem with a pair of tiny leaves and a growing point between them. Okay, it has, it comes later than after radicle, and it will have a short stem, okay, with two pairs of leaves and a growing point between them. Two leaves, a growing point like this, it will have like this, and a short stem like this one. You can say this one. This will a point of the between them. These are the two leaves, and this will be the stem. Okay. like this it will look like. The significance of the seed. What is the importance of a seed? Why do we need a seed? It contains the embryo. The seed contains the embryo. Not rather we want the seed, but the plant needs a seed. Why it is so? Because it contains the embryo, which develops into a tiny plant after germination. Okay. It contains the reserve food material, which is used for the nourishment by the embryo during germination. So two things are important, present in the seed, which makes the seed more important, most important. First one is, it contains the embryo, and secondly, it contains the reserved food material. The embryo, which grows into a tiny plant and after germination, and the reserve food, which is present there, that is used by the embryo to get the nourishment during germination. When during germination, it requires some nourishment, it requires some food for the plant to grow. So that food is obtained from the embryo. That food is present in this seed. Okay. Now, what is the significance of the fruit? Fruit, it protects the seed, and also it helps in the dispersal of the seed. Okay. Next, we see the structure of a maize grain. Okay. We come see the structure of the maize grain. See, this is the structure of a maize grain. I told you when it is a grain, that means the seed coat and the fruit wall that are fused together, which is known as, okay, it is the fruit wall and the seed coat that is fused together. Okay, and it has a protective function. So here we can see first, which we see in this picture. See the first layer that is known as pericarp, which has the seed coat also fused together. The pericarp and the seed coat that are fused together, which forms a single protective layer. Okay. Next come, after the pericarp, there is the aleurone layer. It is the outermost endospermic layer, which is rich in protein. Okay. So the first layer, that is the protective layer, single protective layer, that is the fused pericarp and the seed coat. Okay. After that, inner to that is the aleurone layer. Okay. That is the outermost endospermic layer. That is, endosperm is covered by a layer that is known as aleurone layer, which is rich in protein. Clear? Then comes the endosperm. I told you that endosperm is a covering, which is known as aleurone layer. So below that aleurone layer will come the endospermic layer. Now, endospermic layer or endosperm, which is the major part of the grain, which is rich in starch. Clear? Then comes the epithelium. Okay. See, the endosperm and the this portion, that is the embryo portion. Okay, the embryo portion, the endosperm and the embryo portion. Two portions are major portions in this maize grain. First is endosperm, that is the major part of the grain, and then the lower portion is the embryo region. And the endosperm and the embryo region is separated by an epithelial layer. Okay, this layer separates the endosperm and the embryonic part. Okay, now this embryo, the first portion towards the broader side, you can see, you will find the scutellum or the cotyledon. Okay, the scutellum or the cotyledon, which is above the coleoptile. Scutellum or cotyledon, which lies above the coleoptile. And what is coleoptile? Coleoptile is the protective sheath which covers the plumule. What is plumule? Plumule, I told you, it is the future shoot of the embryo. So that will grow later on into the shoot. Okay, shoot system. So that is covered, that is protected by the coleoptile, and above the coleoptile lies the cotyledon or the scutellum in case of maize grain. Okay. Then below the plumule lies the radicle. Towards the pointed end, we will find the radicle, which will later grow into the root system. And that radicle is protected by coleorhiza, which is also a protective sheath. Okay, so this coleoptile, plumule, radicle, and coleorhiza, all the four constitute what? The embryo. Okay, this picture is of the longitudinal section of the maize grain. So clear about the picture. So first comes the pericarp and the this one seed coat, which is fused together. Okay, that's why it's called a grain. As these two are fused together, that's why it's called a grain. Then lies the alluron layer, which is the outermost this one endospermic layer. Okay, then comes the endosperm that contains the rich in starch, that contains starch. Okay, and then comes the embryonic layer, which is separated by the endosperm is separated from the embryo by a epithelial layer. Then in the endospermic layer we'll find the cotyledon, which is known as scutellum here, and then in the embryo first the plumule is covered by coleoptile, then comes the plumule, then the radicle, which is again covered by coleorhiza. Clear now? Next we are coming to the external feature of the maize grain. See, there is a dotted-like structure, which is the remaining of the style, where the style was attached to it. When it disintegrated, that portion, that scar is there, which is the remain of the style. Okay. Then you can see this is in the middle, the dotted structure. Then the endosperm layer we can see. And in the middle, we will see the position of the plumule and the radicle, just one above the other. The plumule is above, radicle is down. And on the side, there's a mountainous type structure is there that is known as scutellum. Okay. So this is the external feature of the maize grain. Okay. Now the maize grain is a monocot seed. It has only one cotyledon. Now we come to the bean seed, structure of a bean seed, which is a dicot seed. Okay. Now here, the testa is separate from the seed. That is, the fruit wall is separated from the seed coat. Okay. First, we see this is the testa is there. The bean shape, it is a kidney-shaped structure. Bean seed is a kidney-shaped structure. One side is a convex, another side is concave side. So on the convex side, we can see the testa, and on the concave side, we see the hilum. What is hilum? Hilum, it is a whitish oval scar. It's a whitish oval scar. What does it represent? It represents the spot where the ovule, which has now become seed, was attached to the ovary wall through the placenta. When the ovule was attached to the ovarian wall, so through the placenta, that portion is, as the ovarian wall has come out. So that portion is, the scar is left. Okay, that white scar is left, and that is known as hilum. Clear? And see this in the middle picture, you can see the portion of the hilum, that oval shaped structure. That is the hilum. Okay, and just above the hilum, just above the hilum, there is a small hole-like structure, which is known as micropyle, which is a tiny pore through which the water and mineral salts get inside the this one seed, inside the seed, and also the pollen tube entered into the ovule. When the seed was not mature, when the seed was not born rather, That time what happened, that when the pollen tube pollination occurred, that pollen tube entered through this micropyle inside the ovule. And later on, this micropyle, through this micropyle, water will get inside the seed. Okay. So this is the marking. And there are two functions of the micropyle. One is the soaking in the water, absorb. I told you it when it is kept in water, it will soak water through this micropyle and make it available for the embryo for germination. Another thing, it provides the diffusion of the respiratory gases from the growing embryo also. Okay, so not only it absorbs water through this micropyle, but it will also exchange the gases for respiration by the embryo, by the growing embryo. Okay, and the third picture you can see here, the seed coat, the testa has been removed here. Okay, and the radicle, we can see a green structure which is coming out, that is the radicle, and the major portion is the cotyledon. Two in numbers are there. Okay, so this is the external structure of the bean. Now we come to the internal structure of the bean seed. Okay, now first we will see the two leaf-like structure and a curved portion and a pointed structure which is coming down. Okay, the two leaf-like structure, rudimentary leaf-like structure, that is the plumule, which forms the shoot system later on. Then the plumule and the pointed end towards the end. Okay, that is the radicle. Just towards the end, you can see that is continuation, which is coming to the end. The pointed structure is the radicle, which will later form the shoot system. Now, in between the leaves and the pointed structure, there's a curved wide portion. Okay, the curved wide portion which is attached to the plumule, that is epicotyl. And just below the epicotyl lies the hypocotyl, which is attached to the radicle. Okay, now what is epicotyl and what is hypocotyl? Epicotyl is the region of the axis between the point of attachment of the cotyledon and the fibula. Okay, so it is the axis between the point of attachment of the cotyledon and the plumule. Plumule and the cotyledon is attached by a axis, which is known as the epicotyl. Clear now? And hypocotyl is the region of axis which is below the cotyledons. Okay, here the plumule is attached to the cotyledon. That axis is known as epicotyl, and the region of the axis which is below the cotyledon, that is known as hypocotyl. Below the cotyledon. Okay, that is hypocotyl. Clear? Now we come to testa, which is known, what I was talking about testa in the other figures. Okay, other images I was talking about testa. What is that testa? Testa is the outermost hard covering, which is brown in color. Okay, it protects the delicate inner parts of the seed from injury and the attack of the bacteria and fungi and insects. So it is the outermost hard covering of the seed, which is brown in color. The outermost hard covering of the seed, which is brown in color, is known as testa. So what is its function? It protects the delicate inner parts, okay, of the seed from injury, from the attack of bacteria, fungi, or any other insects. Clear? Now we come to the testa is the outermost covering, and below the testa lies the tegmen, which is the inner layer, and it has the function of protection. Okay, so inner layer, that is also a seed coat. That is also a layer which covers the seed, but it lies below the testa, and that is known as tegmen. Tegmen, T-E-G-M-E-N, tegmen. Okay, tegmen, it has the function of protection. It protects the seed. Okay. So this is the structure of the bean seed. Clear? Okay, and these two are the cotyledons. Okay, so bean seed, which is cut open to show the various parts. Okay, now what is the difference, major difference between a bean seed and a maize grain? Okay, so bean seed has two cotyledons and maize grain has only one cotyledon, and that is also known as cotylon. Okay, bean seed has no endosperm, maize grain has a large endosperm. Bean seed, the plumule leaves folded, but maize grain plumule leaves are rolled. Okay, bean seed has large plumule, maize grain has very small plumule. Bean seed has visible hilum and micropyle, but maize grain no visible hilum and micropyle. Bean seed has seed which is separately contained in the fruit called pod. Okay, bean seed, they are separately in a fruit, separately contained in a fruit which is known as pod, but maize, the seed wall and the fruit wall, both of them are fused together, forming a grain structure. No seed separates seed. Okay, and bean seed germinates in epigeal. The germination is epigeal germination, and in case of maize grain, it's hypogeal germination. We'll later discuss this epigeal germination and hypogeal germination. Will come later, okay? So next is, we are coming with the difference between monocot and dicot seeds. Monocot seeds have a single cotyledon, dicot seeds have two cotyledons, as the name suggests, okay? Then monocot seeds, leaves have parallel venation, whereas dicot seeds, the leaves have reticulate venation. In the leaf structures where we have studied venation, where we have seen the parallel venation and reticulate venation. Parallel venation says all the veins are parallel to each other. They will not cut one another. But in dicot, they will cut one another, forming a network, which is known as reticulate venation. Then monocot seeds, they have fibrous roots, but in dicot seeds, they have taproot system. Okay. Then monocot seeds, they have a trimerous flowers, that is calyx, corolla, or perianth. They are in 3 in numbers or multiples of 3 in numbers. Okay, excuse me. And in case of dicot seeds, their flowers will be like pentamerous flowers. That is calyx, corolla. They will be in 5 or the multiples of 5. Okay, like the petals will be 5 petals, calyx, that is the sepals will be 5 calyx, 5 sepals, like that, or multiples of 5. And in trimerous, the calyx, the sepals will be 3 in number, or 6 in number, or 9 in number, like that, multiples of 3. Okay, now we come to germination. What is germination? Germination is formation of a seedling which is developed from the embryo. Seedling formation which is developed from the embryo. That is the embryo developing into seedling, that is known as germination. Okay. Now, the seeds which contain the dormant embryo, we know. The seeds contains what? Dormant embryo. Embryo which is sleeping inside the seed. Now, under dry conditions, the embryo will be inactive. Okay, that is the dormancy state, you can say. Outwardly, from outward appearance, it will be that it is dead. No, no chemical, no action is being taken place from the outside appearance. But inside, the chemical activities are going on, and to attain some physiological maturation, chemical activities goes on. Okay, and as soon as good circumstances or suitable circumstances are available, it will start growing into a new plant, that is, it germinates. Okay, now major 2 types of germination are there, that is hypogeal and epigeal germination. But another type of germination is also there, that is known as viviparous. Okay, so first we will discuss the hypogeal and epigeal, and later on viviparous. So what is hypogeal germination? Here, the cotyledon remains underground. Okay, in hypogeal germination, the cotyledon remains underground, and epigeal germination, cotyledon is pushed up above the ground. Okay, in hypogeal germination, epicotyl elongates, and epigeal germination, hypocotyl elongates fast. Okay, in hypogeal germination usually occurs in monocots, monocotyledonous plants, that is, yes, monocotyledonous seeds. Okay, for example, pea and grains. And in epigeal germination, it occurs generally in dicotyledonous seeds, that is, castor and beans. Okay. Now, I told you what is epicotyl and hypocotyl. Epicotyl is the axis which is connecting between the plumule and the cotyledon. So that elongates, that axis elongates, pushing up the plumule with the cotyledon comes up. Sorry, cotyledon will remain inside. The cotyledon will remain inside. Only the epicotyl will elongate. But in epigeal germination, what happens? The hypocotyl elongates. Hypocotyl is just below the axis, which is below the cotyledon. That elongates, pulling the cotyledon above the ground. Okay. So this is the difference between epigeal and hypogeal germination. Okay. So you have understood, I suppose, that what is hypogeal and epigeal germination. Okay. Then we come to what are the conditions which are necessary for germination? 3 conditions are necessary for germination. One is water, temperature, and oxygen. Okay. So water, which is absorbed by the seeds. We know that when the seed is placed in water or in the soil, through the micropyle, the water and mineral salts from the soil get inside the seed. Okay. And that is used by the seed to swell and rupture the seed pod and allow the radicle to come out to form the root system. Okay. And also this water is also, there's another use of this water, that is the growing embryo. Inside the growing embryo, the certain chemical reactions takes place. Okay. And some enzyme action are also taking place on the food which is present in the cotyledon or the endosperm, in the whatever the food is there, that is inside the cotyledon or endosperm. That enzyme action and the chemical reactions which are taking place inside the embryo, growing embryo, that is also done by this water. So that this the food which is present in the cotyledon and endosperm, that is dissolved inside this water, so that the embryo can absorb it and get the nourishment to grow. Clear? Next is suitable temperature. Suitable temperature means what? Optimum temperature, you can say, which is between 25 degrees centigrade to 35 degrees centigrade. That is necessary for a plant, for the plant to germinate, from the seed to germinate. If there is a very low temperature, then the embryo growth will be inhibited. Okay. And if there is very high temperature, so that will destroy the tissues, delicate tissues of the embryo, and the embryo may die. Okay. Certain tropical plants that require some higher temperature than the temperate ones. Okay. Next is oxygen is required for germination. So from air, oxygen is absorbed. Why oxygen is absorbed? For respiration of the plant. That is for oxidation of the food, so that the embryo gets the energy for rapid cell division and the cell growth. Okay. So oxygen is required for the respiration, which is absorbed from the air, and also from the dissolved air inside the soil also. Some air is also there, but mainly it is absorbed from the air, so that the respiration takes place and there is oxidation of food, and the plant gets, and the seed gets, rather the embryo gets energy to rapidly divide its cells and grow. Then seeds sown in very deep in the soil, they will fail to germinate. Why? The seeds which are sown very deep inside the soil, that fail to germinate. Why? One, there are two reasons for that. One is no proper oxygen supply there. It does not get proper oxygen supply for respiration, and the hypocotyl or the epicotyl will not be able to push, rather break and push above the upper layers of the soil. Okay, so insufficient pushing force by the hypocotyl or the epicotyl to break through the upper layers of the soil. That is, it will not be able to break the upper layers of the soil to come out. Okay, that force is not available if it is very deep inside the soil. Epicotyl and hypocotyl will not be able to break the layers of the soil to come out. Now, sir, there are certain experiments to show that the conditions for germinations, the three conditions of germinations are required for, the three conditions are required for the germination. So first, that is, the first is water. Now, we take two beakers, excuse me, beaker 1, beaker A, beaker B, okay, in which we keep certain seeds, gram seeds on dry cotton wool, and another beaker, in beaker B, we take moist cotton wool with some seeds. So after some time, we will see that the seeds which are kept on the moist cotton wool start germinating, but on the dry wool, it will not germinate. This shows that water is required for germination. Next is suitable temperature. See, again here, we keep two boxes. Okay, in one, we keep some ice cubes and we take a beaker in it, keeping some seeds, gram seeds. Okay, and another beaker B, we take some cotton wool, wet cotton wool, and keep some gram seeds. After some time, we will see that the beaker which had ice cubes, the box which had ice cubes, the beaker which was inside the ice cubes, that those seeds will not germinate because that's very cold. But in the wet cotton wool that is kept in ordinary temperature, that's just those seeds start germinating. So this shows that suitable temperature is required for germination. Okay. Even in the hot also, if we keep it in very hot temperatures, the tissues will die. Okay, and the seed will not germinate. Okay. Next is oxygen is required for germination. In this case, what we'll do, we'll take two flasks, conical flasks, okay, and in one flask we keep cotton wool. Moist cotton wool is required that we keep, some seeds we give inside, and in one of the flask we will keep plain water, okay? And we will close both the flask, and in another flask we will take some alkaline pyrogolic acid in a test tube. In both we will keep water and alkaline pyrogolic acid in two test tubes and hang it inside the flask, okay, and close the flask with the cork. After some time, we will see the seeds which were soaked in the cotton wool and the water was there. There the seeds start germinating, but in the other flask where we had kept this alkaline pyrogolic acid, the seeds have not germinated. Why? Because alkaline pyrogolic acid will absorb the oxygen which was present in the flask. Okay, and the flask is devoid of oxygen. It has no oxygen present in it, and so the seeds have not germinated, even though there were the water was there, suitable temperature was there, but still there is no oxygen, and that's why the seed has not germinated. So that this shows that oxygen is required for germination. Next is the 3-bean experiment. Here what we do, we take a beaker with half with water and we keep a glass slide in it in an angle where 3 beans are attached at 3 layers. Okay, one will be at the lower portion of the slide, which is deep inside the water. The second one will be in the middle, which will be half inside water and half outside. And the third one will be out of water towards the rim of the beaker. 3 beans are attached at 3 portions of the slide. Okay, and we will keep this in a suitable temperature. Okay, after some time we will see that the seed which was deep inside the water, that has started germinating, but only a small portion of radicle has come out. Okay, the middle one, which was half in water and half outside, that has started germinated with the small shoot has come out, and that small radical has also come out. But in the third one, which was outside the water, that has not germinated. So what this shows, this experiment shows that water is required for germination. From the first experiment, water was required, but as there was no oxygen present, so it has not germinated. Very little oxygen was there, which it has used up, so it will not germinate. Okay, it's not germinated properly. It's only a small radical has come out. As some oxygen was there in the water, it has used up, and water was there, so it has small radical which has come out. The middle portion, both the shoot and the radical has come out because it has got water as well as air. Okay, as it has been kept in suitable temperature, so temperature is also there. But in the third one, it is outside the water level. Okay, so water is not there, only air is there, and also suitable temperature is there, but water is not there. So something is missing. Okay, any of the condition, if it is missing, then it will not grow. Okay. Now, I told you there was continuation in the 10th point. There was 3 types of germination. I told you hypogeal, epigeal I discussed, viviparous I had not discussed. So I told you viviparous germination I will discuss later. So what is viviparous germination? Vivipary. Okay, viviparous germination has come from the word vivipary, which means producing living young ones. Okay, producing living young ones is viviparous germination. The seed germinates inside the fruit while still attached to the parent plant. Okay, the seed germinates inside the fruit while still attached to the parent plant. It's still attached to the parent plant. At that time only it will start germinating. After which the plant drops the seedling into the soil. Okay, after germination, it will drop the plant into the soil, and where it will develop its root system, which is known as rhizopora. Okay. Now the seedling has grown. After the seed has dropped in the soil, the plumule has come out, and the radical has come out. Okay, the plumule will go into the grow into the shoot system, and the radical will grow into the root system. So germination ends up into seedling. Okay, germination ends into seedling. Now the seedling, it has stages from a seed before it has become fully independent of the food stored in it. Oh, seedling is the stage from a seed. From a seed, it goes to seedling. So from that stage, it becomes a seedling before it has become fully independent of the food stored in it. So till it has used up all the food and it is independent from, it can independently create its own food. Okay, so till then it is known as seedling. after it has come, after it has been independent from the seed of the end of the food which is stored inside it, it will become the plant. Okay, small plant it will form. So from that fully independent means roots absorbing water and minerals from the soil, leaves start manufacturing food. So till that stage, it is seedling. Okay. That then after this happens, that roots absorb water from and minerals from the soil, leaves start manufacturing food. Then it will become mature, producing flowers and seeds. Okay, so this is the cycle which happens in the seed structure after germination. Okay, so this is the end of the chapter. If you have any doubt, any question, you solve those questions, whichever questions you get on this chapter. If you have any doubt, please do write in the comment box. I will reply you as soon as possible. You can also write in my email ID that is kasturi74@gmail.com. I will reply you as soon as possible. Okay. So till then, bye bye, and do meditate before you start your day, and don't forget to subscribe my channel and ring the notification bell. Thank you.